Okay, so now we have kind of talked about the semi-conservative model. So now let's go through the molecular mechanics of how DNA replication occurs in prokaryotes and eukaryotes. We'll talk about the similarities, and then we'll get into a little bit of the differences that comes from the different ways that prokaryotes and eukaryotes pack their DNA. All right. So I basically smooshed section 4 and 5 of chapter 14 together into one kind of go, so be ready for kind of a longer video. So we'll start with DNA replication overall, and then we'll go into the comparing and contrasting of prokaryotes and eukaryotes. And again, this is a link to this PDF version of this book. So we can start talking about using this as an extra resource when we're talking about DNA replication. So definitely check that out, because it's a really useful way to learn these mechanisms. Okay, so when we talked about Sanger sequencing, I introduced DNTPs. Again, so we use these in our bodies. You're using them right now. I'm using these right now as our cells are replicating. So DNTPs, again, means the D comes from the deoxyribose. So that's the same D that gives DNA, it's D. The T is tri, and the P is phosphates. So what these DNTPs provide is the nucleotides, which, remember, are the monomers or the building blocks of our nucleic acids. But because of that energy that's stored in those 3 phosphate groups, we're actually providing also the energy that is needed to do that dehydration reaction to link those nucleotides together. So that's all handy dandy, that the energy that's needed and the building blocks are all packed into one tidy little package that we call DNTPs. Okay. So basically, what happens during DNA replication is we gotta unwind the DNA double helix. We have to open up those, create those double strands into single strands, and then we gotta prevent it from closing up again. We do that in 2 different ways, and then we're gonna say, okay, this is where copying is gonna start, and then we actually do the actual copying of the DNA. And then remember that DNA is anti-parallel, which causes problems in DNA replication because there's only copying only happens from 5' to 3'. And so there's harder things that we got to deal with. And then in order, when I say clean up the mess for step number 8, it means getting rid of the RNA primers. That'll make more sense later. And then we have to make sure that everything's correct, because we don't want to make mistakes as we're copying our genes, because that's a bummer. And then we got to close everything up again and tidy it up, and then we're donesies. Okay, so let's go through these steps. Oh, no, let's not go through the steps. Let's talk about the major players. This is the starting lineup. I don't know about sports, but I guess LeBron James is like a basketball guy who's like really good, I guess. And so like in basketball, there's like 5 players per team on the court at a time, right? And so that's conveniently, that is how many enzymes, so like major classes of enzymes that are included in DNA replication. So we can talk about these enzymes using a basketball analogy. And so these different, there's the supporting players, but then there's the star, there's the LeBron James, and that is our polymerase. So let's go through these guys and talk about what they do real quick. So we've got our topoisomerase, And his job is just to unwind stuff. We got helicase, and that job is to unzip boop, 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 the DNA molecule so that we can actually do that replication, that semi-conservative replication. We got ligase, which tidies everything up, links everything together nice, and then we have the primase. So this is kind of like the number 2 star. This tells us where to go, where the copying actually starts. And then the polymerase is the actual enzyme that does the actual copying. And so if you'll notice that these ases are all enzymes, and so usually enzymes have the last name of ase. And so then the first name usually tells us a little bit about what they do. So polymerase is the enzyme that does that creates the polymer. All right, so this is our starting lineup. So let's go through these steps a little bit just to get this whole like concept down about DNA replication. Okay, so step 1, we got to unwind that double helix. So remember, we talked about this twisted ladder analogy. We got to untwist it so we have, and that's what this enzyme does, the topoisomerase, and it makes it a lot easier for the helicase to actually break those hydrogen bonds that keep those. But let's talk about that guy now. So helicase breaks the hydrogen bonds that are holding the bases together. And so there's I think I have in here somewhere a link to one of those crash course in biology videos where he's like, hey, what's the similarities between helicase and teenage boys? Is they both want to unzip your genes. So that's why, so that's how I can always remember helicase, because of that really corny joke that I like. Okay, so that's helicase. Its job is to unzip the genes. And then steps 3 and 4, so those hydrogen bonds want to form again, right? So, but what they do is to prevent that, we have some of these proteins that bind to the sugar phosphate backbone that keep them from closing back up. They're just prevent it from snapping back in on itself. And then this dude again, this topoisomerase is also keeping everything straightened out ahead of this replication fork. And so that's what this is called right here, where the helicase is. As you can see that it kind of forks off as it's untwisting and unzipping your genes, right? So it's keeping everything straight out. And so that also prevents it from just closing in on itself again. And in prokaryotes, we see that this replication fork is moving at 1,000 nucleotides every second, which is much slower in eukaryotes, but still 100 nucleotides per second is still really, really fast. That this goes going zip, unzipping, and then replication is happening. The reason that it's slower in eukaryotes is just a artifact of their more complicated genomes, and we'll talk about that in a little bit. Okay, so how do we know where to start copying? Well, that's where our primase comes in. This is an RNA primer, and it basically signals start here. So in order for the polymerase to be able to latch on, it has to have something to latch on to. And so that's why we have an RNA primer that docks on first. So here we have a primase that says that's linking the RNA primer onto the template DNA. And then that says start here. And then the RNA primers are later removed. And sometimes we'll see in prokaryotes, we'll see that they're replaced with DNA, but in eukaryotes, we'll talk about what happens and why aging happens, because those things can't actually be replicated in eukaryotes. Okay, So now we get to the LeBron James of the DNA replication world. We actually have our polymerase that is actually copying that. And so in here is these little boxy, orangey yellow things. All right, so that's the enzymes that is actually creating that dehydration reaction and linking those new nucleotides on and synthesizing a new polynucleotide. Okay, so the sliding clamp protein holds on, like it makes sure that the polymerase doesn't fall off. And then we have to remember that DNA synthesis is occurring from the 5' end to the 3' end. And so if we remember back to our little sugar molecules where we have the oxygen and then we go clockwise and number them. So if you're not sure what I'm talking about, go back and check out one of the earlier videos so that you guys can stay with me on that. But remember, it's going from 5' to 3'. And so it's just this directionality. But because DNA is anti-parallel, there's some problems that come with this because so here it is. So now we got the leading strand and the lagging strand. So here's our parental duplexers. Here's the strand that we're copying, being shown in blue. And then we have this nice little leading strand. And so the fork replication is working in this direction. And for the leading strand, no problem. You just throw that RNA primer on there. Polymerase is just chugging along. The fork's going, and the polymerase is going, no problems. Totally fine. The leading strand and the replication fork are working in tandem with each other. No problems. And then there's the lagging strand. It's being copied from the 3' to the 5' end, but that's the opposite way of actually how DNA has to be synthesized. So now we got the problem that we have to go a little ways and then wait for this to be opened up a little bit and then throw another primer on there and then go a little bit. So it's like this jerky, and it creates these things that were discovered by 2 Japanese scientists. Luckily, they were married, and so they have the same last name of Okazaki, and so we can call them Okazaki fragments. And so apparently Okazaki is a very common last name in Japan. When I was doing my undergrad, there was a guy by the name of Bob Okazaki, but everyone's like, dude, was it one of your family that discovered Okazaki fragments? And he's like, no, it's the same as like Smith or Jones in the US. Like everybody, everybody's neighbor is named that. So, okay, anyway, back to DNA. So new RNA primers are being slapped down every time that opens up a little bit, enough for this kind of jerky, fragmented DNA replication can go on. So we're having constant boop, 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 RNA primers, which means then that these are disjunct. Okay, and this is slower. This is what limits how quickly DNA can replicate. Okay, so now in order to have sequenced or to have replicated the lagging strand, the jerk strand that's going backwards, there's a lot of these RNA primers on there. And so, excuse me. So now we have to go back and remove those primers and fill it in with DNA. So the nuclease enzymes breaks down the RNA, and they're taken back and they're recycled and either made into mRNA or tRNA or whatever the hell RNA this needs to be. So it breaks down the RNA, but not the DNA. And that's our cleaning up the mess stuff. So removing those primers and then adding DNA nucleotides in there. Okay, and then the next thing that we got to do is make sure that we're filling in those little spaces that are left by those Okazaki fragments. And so here we can see there's a little break here. And so there's another more specific type of polymerase that its job is to go and plop those, fill in those spots and those Okazaki fragments. And then ligase. So now we have some of these nucleotides that are stuck in there, but they're not linked up in their sugar phosphate backbone. And so that's what ligase is going to do, is they're going to come in and just, Lego those. Instead of just sticking them on with a hydrogen bonding, they got to like do the dehydration reaction and link those, the 5' end to the 3' end. And it just kind of seals everything up, make sure that everything's quality controlled, and then we're golden. Now we have 2 copies of the same DNA molecule that we started on 1 copy. All right, so one of the reasons why prokaryotes can go so much faster is because they don't really have to deal with these Okazaki fragments like we do in eukaryotic organisms, because they have this nice circular chromosome, right? So what they'll do is that there's a region in the genome that has a lot of the AT pairings, and remember that those only form 2 hydrogen bonds to keep each other, so it's less energy to break those apart. And so there's an origin of replication. And so now we can have copying happening in 2 directions around the circle. And so we don't really have to deal with the Okazaki fragments like we do when we have a linear DNA molecule. All right, so let's see, is there anything else I want to say? That, yeah, so this continues around and around the circle until we boop, knock into each other, and then we know we're done. And then those 2 chromosomes can move into the 2 daughter cells during cell replication. Now, of course, eukaryotes are more complicated because, first of all, they have larger genomes. There's more DNA that needs to be replicated. In our bacterium E. coli, there's about 4.6 million base pairs, right? That's a lot. But compared to us humans, then we have about 3 billion base pairs, and some ferns can have 148 billion base pairs in their genomes. That's huge. And not only so, and remember in our prokaryotes, we just have 1 circle, but in eukaryotes, we have lots of linear chromosomes. And then we have this other thing, these histones. Remember, these are the proteins that the DNA coils up around. We got to get those out of the way, and then we got to put them back after we're done making copies. So there's just a lot more steps involved in a eukaryotic organism, and then we do have to deal with those stupid Okazaki fragments. We also see that in our linear chromosomes of eukaryotic organisms, we have more origins of replication. So we might have 1 strand of DNA that is actually undergoing replication at multiple points. Humans can have like 100,000 origin of replication spots. There's more kinds of polymerase in eukaryotes than there are in prokaryotes, but for our purposes for this basic class, we just need to we don't need to know all of the various different kinds. Just know that it's more complicated in eukaryotes, of course. Okay, and so I hinted at this before. So telomers are basically the margins of the chromosomes. And so these are the very ends of the chromosomes that don't get copied during cell replication, during DNA replication. Remember, we talked about in the prokaryotes, where the RNA primer was glommed on, that can be replaced with DNA. But if we have a linear thing, we got to start somewhere. And so then Okay, so now the chromosome is shorter, and so now we have to start here. And so now the next time we replicate, we have to replicate here. And so we actually see this loss of base pairs with age just because we got to have somewhere for that polymerase to dock on. And so that means whatever was before it doesn't get copied. And so this is an active area of understanding aging and potentially slowing down aging, is if we can figure out how to regenerate those telomeres, or otherwise make sure that they're copied. Okay, so I'm going to end this video here. So that was a lot. So make sure that you're taking notes. We're going to practice some of these DNA replication principles when I get back from my trip. So if you're overwhelmed and lost and confused, don't worry about it. We'll do some in-class practices soon. Okay, bye.